Hi, my name is Cheryl Brown. I'm an oceanographer at the US EPA here at Hatfield Marine Science Center. Today I'm going to present some important research that we're doing tracking nutrient and bacteria as co-pollutants and identifying sources contributing to pollution in estuaries in our area. Nutrient and fecal pollution is a widespread issue in the United States, impacting water quality in many water bodies. These two forms of pollution often co-occur entering the water body together and can impact the ecosystem in numerous ways. One of the challenges is that there are often numerous sources and it's difficult to tease out the contribution of each. We can improve the effectiveness of restoration efforts by targeting the major sources contributing to pollution in such systems. Excess nutrient and fecal pollution can have many impacts on coastal ecosystems, which often results in economic as well as human health impacts. One system where this is an issue is the Tillamook Estuary, which is located about 80 miles north of Hatfield Marine Science Center. I'm sure many of you are familiar with Tillamook ice cream or Tillamook cheese. Tillamook has a long history in dairy agriculture. The Tillamook County Creamery Cooperative was founded in the early 1900s, and today there are over 100 farms in the cooperative, and it is the largest employer in the county. The Tillamook Estuaries Partnership monitors the water quality in the Tillamook Estuary and puts out a report card on water quality. Excess nutrients often lead to algal blooms and low dissolved oxygen conditions. This report card shows that the Tillamook Estuary is severely degraded due to low dissolved oxygen. In addition, it also shows that improvement is needed for bacterial pollution. In fact, shellfish harvesting in Tillamook Bay is closed for about 100 days per year, resulting in economic impacts. This at times has resulted in conflicts between the shellfish and the dairy industries. Because of these water quality issues, Tillamook Estuary is the focus of our work on co-pollutants. The Tillamook watershed is primarily forested, with human development and agricultural activities located in the lower portion of the watershed that is adjacent to the estuary. There are numerous wastewater treatment plants that discharge into the estuary. Cow pastures are also located close to the rivers and the estuary. And during high rainflow conditions, such as that shown in the lower left, the fields are often flooded, resulting in close connection between activities in the upland and the water body. This shows on the right here some of the sources of nutrients to the system. The system receives nutrients from the forest, agricultural activities, wastewater treatment, non-point sources such as septic systems associated with residential houses, as well as from the coastal ocean. To test our approach for tracking pollutants, we sampled the 5 rivers that flow into the Tillamook Estuary. For each river, we sampled upstream in the forested part of the watershed and then downstream in the vicinity of human activities. For example, in the vicinity of agricultural activities, in the urban area, as well as in the vicinity of wastewater treatment facilities. We sampled from bridges as well as wading into the streams. We sampled monthly during all sorts of weather conditions, from sunny days to sudden spring hailstorms, such as shown below in the video. When we return to the laboratory at Hatfield Marine Science Center, we filter the water samples. We then take the filter and extract DNA on it, and then we use the DNA markers that are associated with specific hosts to track the sources of fecal bacteria that might be present. We have markers that can detect and quantify the presence of cow, ruminant, human, bird, and dog fecal bacteria. We then analyze the filtrate that passes through the filter for dissolved inorganic nutrients, as well as the stable isotope of the nitrogen present. In our system, nitrate is the dominant form of nitrogen in many coastal systems in our area. The nitrogen in nitrate can occur as 2 different stable isotopes. Stable isotopes are naturally occurring forms of the same element with differences in their number of neutrons which influence their mass. Nitrogen from specific sources often have a specific stable isotope signature or fingerprint. For example, animal and human waste usually has more of the heavier form of the nitrogen present. On this slide, we're going to walk through how we can use microbial source tracking, nitrogen stable isotope, combined with watershed models to identify the sources contributing to fecal and nitrogen pollution in this system. For each sampling location, we used a watershed model to estimate the agricultural contribution to nitrogen loading at that location, and that is shown on the x-axis. The y-axis value of each point corresponds to the nitrate stable isotope signature. We can see as the agricultural contribution to nitrogen loading increases, the stable isotope of nitrate increases as well. We then colored each symbol by the amount of ruminant marker present. As the agricultural nitrogen input increased, we detected more ruminant marker. In Tillamook, the ruminant signature is primarily associated with cows. So blue indicates a stronger cow signature, while red indicates a lower cow signature. The size of the symbol is scaled by the amount of human marker detected. So all of the ones that fall along the line either had non-detectable or low levels of human marker present. There were 2 stations with isotope values that were elevated, as well as elevated human marker, and those were located in the vicinity of a wastewater treatment facility discharge. This shows how we can use these different tools to identify the sources of nitrogen and bacteria to the system. So what did we find in this study? We found that nitrogen and bacteria are entering the Tillamook tributaries as co-pollutants, meaning they are entering together. We found that by combining microbial source tracking and stable isotopes, we can identify the sources. Nitrogen and bacteria were elevated in the proximity to agricultural activities. We also found that cows were the cause of this downstream elevation of nitrogen and bacteria. This isn't surprising considering that there are more cows than people in Tillamook County. The one exception to this pattern was downstream of wastewater facilities, which had a waste signature associated with humans. For more information on this research, you can email me at the address provided. For more information about nutrient pollution or what you can do to help prevent pollution, please go to the links below. Thank you.